Okay, so thanks so much for hanging in there towards the end of the day. I'm very excited to present. These are very preliminary new findings out of a study that I've been working on with Erica Forbes, who's here, and Kate Keenan and Allison Hipwell, along with my trainees, that is focusing on understanding development of depression in a sample of girls that's been studied for a long time, so I'll go into that in a bit. So this is an exciting chance to sort of share some of the things we're starting on and get some feedback and answer questions. So as I said, one of the goals of this project is to begin to understand brain and behavioral manifestations of maladaptive emotion expression and regulation in girls. And so starting with trying to understand, well, what are some of the precursors we see when girls are younger, late childhood, that relate to, there show evidence of early depressive symptoms, evidence of emotion dysregulation. And of course, as a developmental psychologist, seeing how this unfolds over time and continuing to track depression symptoms, emotion dysregulation, these different dimensions of affective behavior. And I'm just showing a limited set of constructs that are relevant. There surely are others to add. But added to this picture is trying to understand the role of the brain as an outcome, as a predictor in some of these associations. And, so in particular, taking a focus on structural variation, as well as function during emotion processes. And trying to understand, are there altered maturational patterns that we might see in brain development? And in doing so, because we know that adolescence is this very vulnerable period for the neurobiology underlying affective behavior that we see. So we've been addressing this sort of overview picture in the Pittsburgh Girls Study. So the Pittsburgh Girls Study is a community sample of over 2,000 girls who've been studied in the city of Pittsburgh since the age of 5. And when they were 9, Kate Keenan came along and recruited a subset of the girls from the larger study based on elevated mood symptoms at age 8. And so that sample became the Learning About Girls' Emotions sample and is comprised of 232 girls that have been studied since then. And then we came along and introduced a neuroimaging component to that study, where we've just finished studying these girls from the ages of 16 to 19, doing annual assessments that involved neuroimaging. So today I just wanted to highlight some of the initial findings that we've been working on in my lab, particularly with my postdoc Veronica, who's sitting over here. And so it's exciting to start getting more deep into the data. So in terms of depression symptoms, we've been looking at the child symptom inventory, and then as they've gotten older, the adolescent symptom inventory, as well as drawing a measure of emotion regulation that was collected at multiple time points about sadness management, the children's ability to manage their feelings of sadness. And then I'm going to focus on some structural data that we've collected just from the age 16 assessment, but we have it at 16, 17, 18, and 19 now, so I'm excited to hopefully we can have some discussion a little bit about doing that kind of longitudinal analysis with neural data. I'm also going to share with you some functional imaging findings, specifically looking at age 16 to 17, where we're focusing on a sadness introspection, and I'll say more about that in a second. So one of the first questions that we've been kind of getting into is whether the timing and the degree or the severity to which early depressive symptoms might impact later subcortical volumes. And so in 137 of the girls that we completed through the protocol, we created some regions of interest to assess the volumetric size of them. We did bilateral, so we ended up with 12 regions. And then what we were able to do was to look back at their childhood symptoms. And so in this, oh, my slide got a little mushed. In this slide, we have found some differences that are specific to thalamus. So right thalamus is showing this difference. Specifically when we see high levels of severity in depression symptoms when the girls were ages 5 to 9. Okay, so we're sort of averaging across that age group. And so those girls who've had the higher levels of severity within that period of development are showing a larger thalamus at that age. When we looked at ages 13 to 15 in terms of severity, we're seeing a difference with left nucleus accumbens. And so that's the orange bar over there. So the girls who've had the highest severity, ages 13 to 15, are showing a larger left nucleus accumbens relative to those kids who had moderate or lower severity during that age. We did not see any volumetric differences in the age range of 10 to 12. And so we're just beginning to look at this, and it's giving us some ideas and thoughts about perhaps some clues about the timing of when emotions might be sort of more or less dysregulated, when in development that might be important, and how that might relate to certain regions within subcortical networks. A second question that we've been pursuing involves the functional. So in this other study that we're working on now, we've posed the question, does change in depressive symptom severity relate to change in ventral medial PFC activity during this process of sadness introspection? And so given the disorder and the symptoms, we started this study with this, with some thinking about, how are girls responding to stimuli that are depicting, sad emotions, and asking them to reflect on the stimuli that they see with this question of how sad does this person make you feel? So we're sort of starting to think about this as sort of a turning attention inward in terms of sort of an introspective questioning about how this kind of sad stimulus makes you feel sad, relative to a more neutral, non-emotional kind of question, how wide is the nose? So this is essentially our main contrast that we've been looking at in our MRI analysis. And we've been focusing on ventral medial prefrontal cortex, in part given its involvement in a range of studies in self-referential processing. There's evidence for increased activation here in relation to negative self-focus, change in depression symptoms, as well as increased activity when an individual relapses after having remitted as well. So it seems, it's a relevant area of interest to focus on. And so our initial results shown here, on the left we have highlighted with the green circle a cluster that emerges when we just look straight at that contrast in the ventral medial PFC. So this increased activation is in response to how sad versus how wide while looking at sad faces. And we've extracted the data from that cluster as well at 16 years of age, as well as at 17. And so the graph on the right shows you the change in that activation from 16 to 17 against the change in depression symptoms from 16 to 17 as well. And so you can see that as there's a greater change in symptoms, we are also seeing greater change in this area of activation specific to this contrast. So this might have some implications for perhaps for treatment. If this is an area that's sensitive and more changeable, it might help us figure out some specific targets in terms of how we help youth focus their attention, or what are they focusing their attention on in some of their depressive thinking and so forth. Finally, just to push that a little bit further, we wanted to understand, again, as I mentioned before, some of these early precursors of depression. And so in particular, drawing on that sadness management scale from when the girls, here we've used it from ages 9, 10, and 11. So posing this question of, do early patterns of sadness inhibition when they were 9 to 11 predict change in depressive symptoms later? And do we have some evidence for the role of vmPFC in this relationship? And so here you can see the association between that greater inhibition of sadness, so sort of suppressing those sad feelings based on some self-report data. Hopefully across these ages, it's helping us to represent a trait sort of style of their responding, predicting greater change in depression symptoms from 16 to 17. When we include the change that we saw from 16 to 17 in ventral medial PFC, we can see that it mediates the relationship between that early sadness inhibition and later change in depression symptom severity. So